Hello everybody, I am Dr. Sumati from the Department of Biochemistry, Biotechnology and Bioinformatics of Avnashlingam University. I am taking modules under the Nutritional Biochemistry paper. In the present module, we are going to discuss about the metabolism of carbohydrates, that is glycolysis, TCA cycle and electron transport chain, and we will also be discussing about the energy aspects involved in these three pathways. Let's move on to the topic of carbohydrates. Having seen how important carbohydrates are in acting as energy source as well as structural elements of the body of both plants and animals, now we shall see how these carbohydrates are metabolized and yield energy to fuel various metabolic processes. The first step in the oxidation of glucose is glycolysis, where it is oxidized to yield pyruvate and ATP. Then it enters into the TCA cycle, where pyruvate is oxidized to carbon dioxide and NADH is produced, and also various important metabolic intermediates are produced. Finally, these electron carriers enter into the electron transport chain, where it converts redox energy available from the oxidation of NADH and FADH2 into a proton motive force that is used to synthesize ATP through a process called oxidative phosphorylation. See the objectives of this module. To understand the pathway of oxidation of glucose molecules to pyruvate. To learn how the products of glycolysis feed into the citric acid cycle and how the products of the citric acid cycle ultimately end up with the products of glycolysis in the electron transport chain. To elucidate the general flow of electrons from glucose to NAD plus and FAD plus through a series of chemical intermediates. To explain how ATP is produced through oxidative phosphorylation. So, the summary of the talk will be how glucose is oxidized to two molecules of pyruvate, and then this pyruvate is converted into acetyl-CoA and that enters into the Krebs cycle, where it is finally oxidized to carbon dioxide and various reducing equivalents, which finally enters into the electron transport chain to yield ATP. We will also see how complete oxidation of one molecule of glucose yields 36 ATP molecules. First topic we will take up will be glycolysis. The word glyco means sugar and lysis means breaking. It is also named as the Embden-Meyerhof pathway. Glycolysis is a common pathway present in all organisms, starting from yeast to mammals. As I told you earlier, the glycolytic pathway describes the oxidation of glucose to pyruvate with the generation of ATP and NADH. In eukaryotes, glycolysis takes place in the cytosol. Now, what's the net result of the glycolytic pathway? Glycolysis is a pathway that starts with glucose and ends with 2 3-carbon units, that is pyruvate, and the released free energy is used to synthesize ATP from ADP and PI. The pathways are interconnected. Let's take an overview of the pathway of glycolysis. Now, before we move on to the steps, another point to consider is, once glycolysis is over, the cells are left with two alternatives, that is, there are two post-glycolysis pathways. This depends on the condition in which the cells are present. As I told you, pyruvate the end product of glycolysis has two different fates. It depends on what the cell needs, what the cell has, and type of cell it is taking place. Aerobic oxidation results in the production of 2 molecules of pyruvate and also produces 6 molecules of carbon dioxide, 6 molecules of water and yields 30 ATP molecules. Now, what if cells don't have oxygen? The cell should have a backup pathway to operate to get energy. So, pyruvate is converted to lactate. On the other hand, anaerobic fermentation also results in the formation of an acid called as homolactic acid in the muscles and results in the production of 2 molecules of pyruvate, which results in the production of 2 molecules of lactate, and no ATP is synthesized. What if it is not my cell but a bacteria or yeast? Then it leads to the formation of ethanol. In yeast, it is converted to 2 molecules of pyruvate, which in turn is oxidized to form carbon dioxide and 2 molecules of ethanol. So, the net reaction will be glucose plus 2 NAD plus 2 Pi plus 2 ADP will result in the formation of 2 molecules of pyruvate, 2 ATP, 2 NADH and 2 molecules of water. Now, let us dive into glycolysis. This figure is the overview of glycolysis showing all the players. The first thing you should know is the name of the structures of common sugars and their derivatives. You should also know the name of the enzymes of the pathway, which tells what the reaction is going on. Let's look into the process. They are divided into two main phases. They are energy investment phase and energy generation phase. So, in the first phase, energy is utilized, and in the second phase, it is regenerated. That is, we get 2 ATPs and also get something else. They are NADH and pyruvate. If it is further oxidized by TCA cycle, we get 38 ATP in one condition, and when there is no oxygen, then it results in the production of 36 ATP. Now, glycolysis occurs in 3 stages. First is the priming stage. The reactions 1 to 3 consisting of a phosphorylation, which is done by a kinase, and isomerization, and a second phosphorylation again by a kinase. The strategy here is to trap the glucose inside the cell and form a compound that can be readily cleaved into symmetrical phosphorylated 3 carbon units. The second is the splitting stage, where the reactions 4 and 5 take place, where the fructose is cleaved into 2 3 carbon units that are readily interchangeable. Third is the oxidoreduction phosphorylate stage, where reactions 6 to 10 consisting of a redox reaction, 2 dephosphorylation reaction, an isomerase reaction and a lyase reaction. In this stage, ATP is harvested when the 3 carbon fragments are oxidized to pyruvate. Net profit is 2 ATPs and 2 NADH per glucose molecule. Now, going to the reactions of glycolysis. The first few steps, as I told you, are energy utilizing phase. Let us take a look at the reactions. In the first step, glucose is converted to glucose 6-phosphate and the first molecule of ATP is utilized, and the enzyme is hexokinase, which transfers a phosphoryl group. It is one reaction which is regulated among the 3 regulatory enzymes. The second step in glycolysis is the transfer of the phosphate group, which is transferred from glucose 6-phosphate to fructose 6-phosphate. The enzyme that catalyzes this reaction is phosphoglucoisomerase. Here what happens is the aldehyde group is converted to a keto group, which is an isomerization. In the next step, fructose 6-phosphate is converted to fructose 1,6-bisphosphate. This reaction is catalyzed by phosphofructokinase. It is the most important regulatory enzyme of the glycolytic pathway. There are several things which will regulate this enzyme. Here again ATP is required. Now, in the next step, a lyase type reaction where the molecule splits in the middle and the reaction is catalyzed by the enzyme aldolase. It is split into dihydroxyacetone phosphate and glyceraldehyde 3-phosphate. The reaction is a simple aldol cleavage. Last energy investment phase involves conversion of dihydroxyacetone phosphate to glyceraldehyde 3-phosphate. Now, 2 molecules of glyceraldehyde 3-phosphate are formed, and so now onwards 2 molecules of each is present. Now we move on to the oxidation phase of glycolysis. In the energy generation phase, 2 ATP are produced, and because there are 2 molecules of glyceraldehyde 3-phosphate, 4 ATPs are produced. The next step is the first high energy intermediate formation, that is glyceraldehyde 3-phosphate is converted to 1,3-bisphosphoglycerate, and the enzyme is glyceraldehyde 3-phosphate dehydrogenase. This reaction is a oxidation and phosphorylation. The first thing to happen is oxidation, and then the phosphate is put on the acid. The oxidation converts NAD to NADH. The next reaction is conversion of 1,3-bisphosphoglycerate to 3-phosphoglycerate, and the first ATP generation takes place here, and the reaction is catalyzed by phosphoglycerate kinase, and the first ATP is generated. This is a substrate level phosphorylation. The next step is the conversion of 3-phosphoglycerate to 2-phosphoglycerate. Looks like a simple isomerization reaction catalyzed by a mutase which transfers a phosphate group from the 3rd position to the 2nd position. This bisphosphoglycerate is an important molecule as it binds to the central cavity of hemoglobin molecule and stabilizes the T form, that is deoxyhemoglobin. Thus, high bisphosphoglycerate in the blood reduces the oxygen affinity of hemoglobin to the molecules. The next step, 2-phosphoglycerate is converted to phosphoenol pyruvate just by the removal of water and is catalyzed by the enzyme enolase. Finally, the phosphoenol pyruvate is converted to pyruvate by the enzyme pyruvate kinase, and this is the 3rd regulatory enzyme of glycolysis. Here again it is a simple hydrolytic reaction where again one molecule of ATP is synthesized. Now, the reactions of glycolysis are finished. One thing you have to notice is that NADH is formed from NADP plus during glycolysis. In the presence of oxygen, this NAD plus is regenerated by electron transport chain. The redox balance of the cell has to be maintained for the further cycle of glycolysis to continue. Now, what is the fate of pyruvate? As discussed earlier, NADP plus can be regenerated by one of the following reactions or pathways. Pyruvate is either converted to lactate or pyruvate is converted to ethanol. In the presence of oxygen, the pyruvate is converted to acetyl-CoA that enters into the next cycle, that is the TCA cycle, and gets completely oxidized to carbon dioxide. NADH is reoxidized by sending electrons into the mitochondria. Under anaerobic conditions, the NAD plus is replenished by the reduction of pyruvate by two processes, that is by homolactic fermentation that takes place in the muscle and by alcoholic fermentation that takes place in yeast. First, let us see about homolactic fermentation. The overall process of anaerobic glycolysis in the muscle is that glucose is finally converted to two molecules of lactate. Now, much of the lactate in the muscle cells are carried by the blood to the liver where it is reconverted to glucose. Muscle fatigue and soreness is caused by the accumulation of glycolytically generated acid, but not lactate. Now, what happens during alcoholic fermentation? Here again it is a two-step reaction. First, decarboxylation of pyruvate by pyruvate decarboxylase forms acetaldehyde. Then, this acetaldehyde is reduced to ethanol by alcohol dehydrogenase. Both homolactic and alcoholic fermentation have the same function, that is anaerobic regeneration of NADP+. So, as to continue glycolysis quickly to produce ATP molecules. Now, having seen the steps of glycolysis, we should understand the fact that glycolysis is used for rapid ATP production. ATP production of anaerobic glycolysis is approximately 100 times faster than that of oxidative phosphorylation. Thus, tissues such as muscle consuming ATP rapidly regenerate it almost entirely by anaerobic glycolysis. Now, pyruvate is converted to acetyl-CoA before it enters into the TCA cycle. Let's see the steps between glycolysis and citric acid cycle. Now, before we can get to the next stage of cellular respiration, that is the TCA cycle, there is some preparation work that needs to be done. Before you barbecue your steaks at the picnic, you probably want to thaw and season them first to get them ready to go. So, let's get those products from glycolysis ready for the grill. Each pyruvate which is produced in the cytoplasm enters the mitochondria to be converted into acetyl coenzyme A. Remember that two pyruvates are created from one cycle of glycolysis, meaning two acetyl-CoA molecules are produced. Acetyl-CoA is a 2-carbon molecule. This preparation of citric acid cycle is called as pyruvate oxidation. The reaction links glycolysis to citric acid cycle, and the enzyme pyruvate dehydrogenase catalyzes oxidative decarboxylation of pyruvate to form acetyl-CoA. Now, we will move on to the next cycle, the citric acid cycle. It is a central metabolic pathway that completes the oxidative degradation of amino acids, monosaccharides and fatty acids. In aerobic catabolism, these biomolecules are broken down to smaller molecules that ultimately contribute to a cell's energetic or molecular needs. So, the citric acid cycle aids two main purposes. One is to increase the cell's ATP producing potential by generating a reduced electron carriers such as NADH and reduced ubiquinone, and second, to provide the cell with a variety of metabolic precursors. The cycle begins with the formation of a 6-carbon citric acid by the reaction between acetyl-CoA and 4-carbon oxaloacetate. Through the following steps of the cycle, 2 of the 6 carbon of citric acid leave as carbon dioxide to ultimately yield 4-carbon product oxaloacetate that is used again in the first step of the subsequent cycle. During the 8 reactions that take place for every molecule of acetyl-CoA, the cycle produces 3 NADH, 1 FAD or FADH2 along with 1 molecule of ATP. Now, this picture shows the overall reactions and formation of both NADH and ATP. Now, let us see the reactions one by one. The first reaction of the citric acid cycle is catalyzed by the enzyme citrate synthase, where, as I told you earlier, acetyl-CoA combines with oxaloacetate to form citrate. In the reaction 2, the enzyme aconitase catalyzes the conversion of citric acid to isocitrate. Now, two events occur in the reaction 3 of citric acid cycle. The first reaction generates NADH from NAD. This reaction is catalyzed by the enzyme isocitrate dehydrogenase to yield an intermediate which then has a carbon dioxide molecule removed to yield a compound called alpha ketoglutarate. One important thing to be noted is that this compound is very important intermediate yielding many compounds. In reaction 4, the alpha ketoglutarate loses one carbon dioxide molecule and coenzyme A is added in its place. The decarboxylation takes place with the help of NAD that is converted to NADH. Alpha ketoglutarate dehydrogenase enzyme catalyzes this reaction to form a compound called succinyl-CoA. Then, the enzyme succinyl-CoA synthetase catalyzes the fifth reaction of the citric acid cycle. Here, a molecule of guanosine triphosphate, that is GTP, is synthesized. As a result, you get succinate. In the next reaction, the enzyme succinate dehydrogenase catalyzes the removal of 2 hydrogen atoms from succinate to form fumarate. In the following reaction, the enzyme fumarase catalyzes the addition of water molecule to fumarate to form an hydroxyl group to yield the molecule L-malate. And in the last reaction of the citric acid cycle, we regenerate oxaloacetate by oxidizing L-malate with a molecule of NAD to produce NADH. Next, let us see how the reaction of citric acid cycle are regulated. This regulation is very straightforward. 3 factors govern the rate of flux through the cycle. 1. Substrate availability, 2. Inhibition by accumulating products, and third is by allosteric feedback inhibition of early enzyme by later intermediates of the cycle. There are 3 strongly exergonic steps in the cycle, those catalyzed by citrate synthase, isocitrate dehydrogenase and alpha ketoglutarate dehydrogenase. Each can become the rate limiting step under some circumstances. So, finally, to conclude, let us review what the citric acid cycle has generated from one acetyl-CoA molecule. The acetyl-CoA has been oxidized to 2 molecules of carbon dioxide, 3 molecules of NAD were reduced to NADH, 1 molecule of FAD was reduced to FADH2 and 1 molecule of GTP, which is an equivalent of ATP, was produced. Reduction is really gain of electrons. In other words, NADH and FADH2 molecules act as electron carriers and are used to generate ATP in the next stage of glucose metabolism, which is nothing but oxidative phosphorylation. So, in the next part, we will learn what processes take place to ultimately derive the majority of ATP we need to fuel our daily activity. So, the next topic we are going to discuss is the electron transport chain, which is the final and most important cell of cellular respiration. The reducing intermediates produced during glycolysis and the citric acid cycle make the necessary precursors for the electron transport chain, which creates majority of ATP. The electron transport system, also called as the electron transport chain, moves electrons and converts redox energy available from the oxidation of NADH and FADH2 into a proton motive force that is used to synthesize ATP through conformational changes in the ATP synthase complex through a process called oxidative phosphorylation. Now, the citric acid cycle oxidizes acetate to 2 molecules of carbon dioxide while capturing the electrons in the form of 1 molecule of FADH2 and 3 NADH molecules. These reduced molecules contain a pair of electrons with a high transfer potential. These electrons are eventually going to be transferred by a system of electron carriers to oxygen and finally they form water. The process takes place inside the mitochondria and it is a major energy source used to produce ATP by oxidative phosphorylation. Now, this picture shows how many ATPs are produced during these three processes. ATP synthesis is not an energetically favorable reaction. Energy is needed in order for it to happen. And where do we get this energy from? This energy is derived from the oxidation of NADH and FADH by the 4 protein complexes of the electron transport chain. The 10 NADH that enter the electron transport originate from each of the earlier processes of respiration. 2 from glycolysis, 2 from transformation of pyruvate to acetyl-CoA and 6 from citric acid cycle. The 2 FADH2 is created in the citric acid cycle. The electron transport system contains 4 protein complexes. Before we move on to the actual process by which the transfer takes place, we should know the components of the electron transport chain. The first component is NADH. So, we have seen that NADH is generated in the matrix by the reaction of different enzymes like pyruvate dehydrogenase, isocitrate dehydrogenase, alpha ketoglutarate dehydrogenase and malate dehydrogenase. Second one is flavoproteins. So, flavoproteins have either FAD, that is flavin adenosine dinucleotide, or FMN, that is flavin mononucleotide as the prosthetic group. Flavoproteins can accept or donate electrons one at a time or 2 at a time. Therefore, they are often intermediates between 2 electron acceptors or donors and 1 electron acceptor or donor. The third component is coenzyme Q. This is a versatile cofactor because it is a soluble electron carrier in the hydrophobic bilipid layer of the inner mitochondrial membrane. Like flavoproteins, coenzyme Q can accept or donate electrons one at a time or 2 at a time. Next are cytochromes. These are proteins that function as 1 electron carriers. Cytochrome C, C1, A and A3 are the different cytochromes seen. Next is iron sulfur proteins. These are non-heme iron sulfur proteins. They are FES, Fe2S2 and Fe3S4. Next is copper proteins. Copper bound proteins participate in 1 electron transfer involving Cu plus and Cu2 plus oxidation states. The next session we are going to have an overview of the reactions of the electron transport chain. The electrons move along the electron transport chain going from donor to acceptor until they reach oxygen, the ultimate electron acceptor. As we have seen earlier, the components of the electron transport chain are organized into 4 complexes. Each complex contains several different electron carriers. Let us look at them one by one. First, complex 1. This complex accepts electron from NADH and serves as the link between glycolysis, the citric acid cycle, fatty acid oxidation and the electron transport chain. Complex 1 is also called NADH coenzyme Q reductase because this large protein complex transfers 2 electrons from NADH to coenzyme Q. Complex 1 was previously known as NADH dehydrogenase. Now, this complex binds NADH, transfers 2 electrons in the form of a hydride to FMN to produce NAD plus and FMNH2. The subsequent steps involve the transfer of electrons one at a time to a series of iron sulfur complexes that include both 2Fe2S and 4Fe4S clusters. The final step of this complex is the transfer of 2 electrons one at a time to coenzyme Q. Coenzyme Q, like FMN and FAD, can function as a 2 electron acceptor or donor and also as an 1 electron acceptor or donor. The isoprenoid tail of coenzyme Q makes it highly hydrophobic and lipophilic. Therefore, it is the mobile electron carrier. It can diffuse freely in the bilipid layer of the inner mitochondrial membrane. Now, complex 2 includes succinate dehydrogenase and serves as a direct link between the citric acid cycle, that is an integral membrane protein. The complex is composed of 4 subunits. Complex 2 is made up of 3 FES centers, 1 4-FeS4S cluster and 1 3-Fe4S cluster and 1 2-Fe2S cluster. Succinate is bound and a hydride is transferred to FAD to generate FADH2 and fumarate in the first step of this complex. The electrons from FADH2 are transferred one at a time to the FES centers. The last step of this complex is the transfer of 2 electrons one at a time to coenzyme Q to produce COQH2. Complexes 1 and 2 both produce reduced coenzyme Q. This reduced coenzyme Q is the substrate for complex 3. Now, complex 3 transfers the electrons from reduced coenzyme Q to reduced cytochrome C, which is the substrate for complex 4. This complex is also known as coenzyme Q cytochrome C reductase because it passes the electrons from coenzyme Q to cytochrome C through a very unique electron transport pathway called the Q cycle. The reduced coenzyme Q binding site is now known as the QP site. The electron transfer happens in 2 steps. In the first step, 2 protons are released to the intermembrane space. Then, the 2 electron carrier COQH2 gives up its electron to cytochrome C. The cytochrome C is the only water soluble cytochrome. It is a mobile electron carrier which diffuses through the intermembrane space, shuttling the electrons from complex 3 to CUA site of complex 4. Next, complex 4 transfers these electrons from cytochrome C to reduce molecular oxygen to water. Complex 4 is also known as cytochrome C oxidase because it accepts the electrons from cytochrome C and directs them towards the 4 electron reduction of oxygen to the final destination, namely to 2 molecules of water. So, to make 2 molecules of water, it takes 4 electrons and these electrons come one at a time. Now, each of these complexes are large multi-subunit complexes which are embedded in the inner mitochondrial membrane. The reduction of oxygen by complex 4 involves the transfer of 4 electrons. 4 protons are abstracted from the matrix and the 2 protons are released into the intermembrane space. So, as to summarize the events of the electron transport chain, the electron transport chain is the stepwise process of cellular respiration that is responsible for producing water with the help of oxygen we breathe up to 34 molecules of ATP and NAD and FAD, which are to be recycled to be used again by the citric acid cycle and glycolysis. Now, this process happens in the mitochondria of eukaryotes and cell membrane of the prokaryotes. And the last key point to remember is this only happens in aerobic conditions, that is when oxygen is present. If there is a shortage of oxygen, cellular respiration will take an alternate pathway and at the end of glycolysis resulting in the production of lactic acid and ATP. Now, I am sure that this module would have given you a clear idea about the steps involved in these 3 pathways, namely glycolysis, TCA cycle and electron transport chain, and how ATP has been generated and molecular oxygen is also evolved.